This presentation is about the P blood group system. It's one of the our human blood group system. And in this presentation, first of all, I'm going to talk about how and when the P blood group system was discovered. And then I'm going to talk about the genetic makeups of a P blood group and include antigens, phenotypes, and antibodies. And then I briefly mentioned the biochemical structure of the P a blood group system, and then I am going to talk about the clinical significance of different antigens and antibodies. And the last, I'm going to briefly talk about the method for testing. P blood group was discovered by two people, Mr. Landsterner and Mr. Levin, in 1927. Mr. Carl Landsterner discovered human blood group ABO group in 1900. He also was awarded the Nobel Prize for this discovery in 1930. In 1927, Mr. Landsterner collaborated with Mr. Paul Levin. They did experiments and discovered that injecting the human and the primates' red blood cells into rabbits and absorbing the resident antiserum with the selected red cells. So this kind of experiment led them to discover, later we called three blood groups, M, N, and P antigens. And also later, similar antigens, similar to P antigens found in nature, such as bird droppings, pigeon eggs, hydrogen cyst fluid. Um, P blood group system of human blood composed of like three different antigens on the surface of red blood cells. They are called P, P1, and PK. Those three antigens has five phenotypes: P1, P2, P1K, P2K, and P. The most common phenotype of those five is the P1 phenotypes, which display all three antigens, P, P1, and PK. The second most common phenotype is P2 phenotypes, which has P and PK antigens on the surface of red blood cells. Other three phenotypes, P1K, which is composed of the P1 and the PK antigens, P2K, which has only PK antigens, and the P phenotype with no antigens are extremely uncommon, and they are rare. So for the genes coding for the P antigens, they are on chromosome 22. There are two genes. A4GALT stands for alpha 1 for galactosytransferases, and B3GALNT1 stands for beta 1, 3, and acyl galactose amino transferase 1. So PK antigens is produced by gene A4 GALT, and the P1 antigens are produced by gene B3 GALNT1. So in terms of the biochemical structures of the antigens, they are all on the membrane proteins, and they are all on the surface of a red blood cells membrane. So they the beginning of the sugar chains, are the same, and then later on there's variations. Different antigens has different functions. They all reside on the surface of red blood cells. P antigens um, are receptors for parvovirus B19, which is the agent causing erythema infectiosum, or called fifth disease, that cause transient anemia or plastic crisis. For PK antigens, they can be the receptor for Shiga toxins, and the E. coli associated hemolytic remic syndrome, or HUS. PK antigen also is the receptor for Streptococcus suis, which can cause bacteria meningitis, because it is a bacteria infection receptor. For the P1 antigen, it's the receptor for P fibrated rhopathogenetic E. coli. It's another stream of E. coli, which mainly cause chronic urinary tract infections. Which also P1 antigen not only appear on the red blood cells, also on the tissue cells, such as at the urinary tract. In terms of those 5 phenotypes, P1 phenotype is the dominant phenotype among the populations, seen in 95% of blacks and 80% of whites. The second popular phenotype is P2, which is seen 5% of blacks or 20% whites. Other 3 phenotypes, they are very rare. For example, a very strong anti-PP1 PK antibodies can be associated with delayed DHD and HDFM, but there are absence of P antigens on the red blood cells. P antibodies in the serum has some clinical significance. For example, anti-P1 titers are often elevated in patients with liver efflux or hydatid cyst disease, or for the individuals are frequently exposed to birds, because which the bird drops may have P1 like substance. Another P antibodies is called anti-P P1 PK antibodies, which compose of a mix of 3 antibodies: anti-P, anti-P1, and anti-PK in serum of those P individuals. Allo anti-P is seen in the serum of a P1K and a P2K persons. They are not naturally occurring and predominantly IgM isotope, but also they can be mix of IgM and IgG. There are very little IgG in it. The antibodies are capable of causing HTR and HDFN in newborns or the fetus. Also, there's a relationship between anti-PP1 PK and early spontaneous abortion. The last antibodies I'm going to talk about is auto anti-P. It's auto antibody of a P specificity. They are seen individuals with paroxysmal cold hemoglobinuria, or PCH. This is kind of this disease is in the body. Auto anti-P antibodies bind to the red blood cells its own patient's red blood cells at a cold temperature, and then after circulating back to the core body, which is warm up, so subsequently induce intravascular hemolysis. The antibodies against the PP1 PK antigens may cause transfusion reactions, and antibodies against the P and the PK antigens may cause severe erythroblastosis, fetalis, or a spontaneous abortion. To detect the antibodies, anti-P anti-P antibodies tests are not usually done as routine laboratory methods, but it's possible to detect them using a test called Donnas-Landsterner test. In this test is performed on 2 vials of blood at 2 different temperatures, 4° and 37°. The results are interpreted as a positive only if after the patient's red blood cells have been incubated at both temperature and both showing subsequently hemolyzed. So as a conclusion, P blood group system is usually clinical insignificant. The routine lab test won't test for the P antigens or antibodies. The 3 antigens are coded by 2 genes on chromosome 22. The antigens is dominantly IgM class antigens. The presenting of P antibodies may cause some disease, but because the P antigens is rare, so usually the transfusion routine won't test for it. That's all. Thank you.